Hello, everyone. I'm Dr. Ali Poyanmer, a nephrologist with Kaiser Permanente San Francisco, and your host today's Club. Thank you for joining us today. I'm excited to announce and moderate today's session on getting ready for Flozins, the 2022 version.
Let's start with a little history, an interesting history going back more than almost 200 years ago when the first SGLT2 inhibitor was discovered, Flozin, which is a glucoside of fluoretin, isolated from the root bark of apple trees by some Belgian investigators in 1835. 50 years later, the famous von Mering showed that this agent caused glycosuria and lowered blood sugar, but because it has very poor bioavailability and lots of GI side effects, it was never developed as a therapeutic agent, largely because it's nonspecific and inhibits both SGLT1 and 2. The sodium glucose co-transporters are members of a family of genes, the SLC5 gene family. They were discovered in the early 1990s, and the SGLT2 gene on chromosome 16q was cloned in 1992, but the modern SGLT2 specific inhibitors, such as dapagliflozin, were developed beginning in about 2012. Now, SGLT, the sodium glucose co-transporter, facilitates the transport of D-glucose along with sodium across cell membranes, and it's energy consuming. Two different SGLT2 are representative of the class. SGLT1 is in the intestine mainly, and SGLT2 is mainly in the kidney, particularly in the S1 and S2 segments of the proximal tubule. Both of these proteins are expressed at very low levels in the myocardium in the brain as well, and that may influence some of their other effects. High ambient glucose concentration in the tubular lumina increases the expression of SGLT2 on the cell membrane. Now, we have currently 5 approved SGLT2 inhibitors shown on this slide. The first one was dapagliflozin, and the most recent is ertugliflozin, which is not yet approved by the FDA in the USA. I'll be talking mainly about DAPA, CANA, and empagliflozin in this talk. Now, under normal circumstances, the SGLT2 results in the absorption of about 90% of the glucose filtered at the glomerulus, such that by the end of the proximal tubule there is virtually no glucose, and no glucose is excreted at normal blood sugar levels. In diabetes, several events occur. One is there is glomerular hypertrophy and increased glomerular filtration rate, so-called hyperfiltration, largely due to afferent arteriolar dilatation and increased glomerular capillary pressure. Along with the increased serum concentration of glucose and the GFR, there is an increased filtered load of glucose, and as a result of SGLT2, marked increases in glucose and sodium chloride reabsorption. This complicated process leads to efferent arteriolar constriction and afferent arteriolar dilatation, with decreased sodium delivery to the juxtaglomerular apparatus. Glycosuria is present, and this also contributes to tubular hypertrophy. When you add SGLT2 inhibitors plus renin-angiotensin system inhibitors, the glomerular capillary pressure normalizes, GFR falls due to the reversal of vasodilatation and the reversal of vasoconstriction. The filtered load of glucose decreases somewhat,